do a lot of biomarker. Yes, I do a lot of biomarker discovery for a lot of different types of diseases, mainly glycomics focus, sometimes a bit more proteomics or glycoproteomics. But I've been struggling a lot with glycomics data. Especially, I did a lot of manual interpretation, and Hector started to, he came to visit our group 2 years ago, roughly, maybe. And kind of as a small side project, we started with GlycoGenius, and it turned out to be a lot bigger. But I'm very grateful and to tell you something more about that in a few minutes. But maybe Hector, you can say a little bit more about yourself.
So, hello everyone. I'm Hector Loponte. So, I started my PhD in 2021, and I started working with glycomics mass spec in my master's in around 2019. And currently, I'm on track to finish my PhD. I already have my thesis ready. I will probably be defending it February next year. But since I always struggled also with data analysis since the beginning with glycomics, I started writing my own scripts in my master's, and I had the opportunity to actually work on it full time during my time at Guenevere's lab. So GlycoGenius eventually came to fruition, basically. And yeah, you'll get to know it a little bit very soon. I hope you like it.
Yeah. So we kind of divided it in two parts. So I will give a bit of a short introduction. Okay, what is actually glycogenous and why should you be interested in it if you're doing glycomics work? And then Hector will actually do the real hands-on show you how it works with glycogenous, how to put your data in there and how to get some results. So as I already mentioned, I put two links in the chats, which you can download the software as well as to find some MZML files, which we will use for the data processing later on. So hopefully you have everything downloaded once we get started. So after I finish my introduction. So let me see if I can share my screen. I think you can see my screen now.
Yes.
Okay, great. So we want to go from data to discovery. And here are again the two links. We also have two QR codes where you can find it. Let me know if you have issues with downloading it. If not, then I will just proceed with what is actually the challenge. Because I think most people work with LCMS data for glycomics. So you get a lot of data, a lot of spectra, and what you really feel like that there might be gold hidden in your data. There might be specific biomarkers that you're looking for a specific disease. However, what I always normally do is that I look at my data, I look at each peak, I look at the spectra and try to identify where my glycans are. But you go from spectra to spectra to spectra, and at a certain point, maybe you or even your computer breaks down because it's too much data to handle. So that is actually where a lot of different software tools come in handy. So we have a lot of different there are tools that are already available. So we have Glyco Workbench, which is very good if you want to generate glycan cartoons or if you want to do an in silico digestion to figure out, okay, how will it fragment? We have Glycombo, Glycoforest, Candy Crunch, GlycResoft, and Grid's Toolbox. And we try to map down what are all the specific features that we're looking for. So we want to have kind of ideally a fully automated tool that can identify and quantify your glycans on an MS1 level, on an MS2 level, and hopefully also have a little bit of a library that you don't need to generate all the time, but also doesn't is a it's not directly a fixed library. So there's a little bit of openness to go and look beyond the borders. And as you can see, all of these features cover a little bit, but they don't cover all the features that we ideally are looking for. And that is where GlycoGenius comes in. So we try to make GlycoGenius in such a way where we can really go from the library to the automatic assignment, identification, quantification, and really have the full data analysis workflow. as illustrated over here. So here we have an asterisk. So at the moment, the integration with the databases is not fully there yet, but it is an ambition that we're striving for. So GlycoGenius is not just built from out of nowhere. We actually took a little bit of the parts that we were looking for and that we really liked from the different software tools that are out there. So I worked a lot with Bruker data. So for the data analysis, I prefer some ways how it shows your data, how you can generate your extracted ion chromatograms. GlycoResolve is very nice if you want to have a look more for the identification of your glycans. Skyline is very good if you want to really look at your extracted ion chromatograms, but also look at the isotope scoring. And then GlycoWorkbench is very nice if you want to have your glycan cartoons in there. So we took a little bit of everything and tried to mix that together, and that ended up into being GlycoGenius. So our motivation was very simple: create an open source software tool, user friendly, that anyone hopefully can use, either you're a glyco specialist or whether you're just starting. And what we start with is MS1 level. So it's focus, friendly, and glycomics first. For this, you need an MZML or an MZXML file, so it should work in theory with every mass spec vendor that is out there. And then within GlycoGenius, what you can generate is a glycan composition library. And in this library, you can add modifications, so phosphorylations, sulfations are integrated, but also if you have a specific tag at your reducing end. And also it allows you to add additional modifications if you have modified it in a specific way that has not been incorporated in GlycoGenius yet. It also allows to do calibration of your M over Z values. So if you know your calibration is off, you can still integrate it internally. And I will show in a few slides how that works. And also what you can do is data curation. So with data curation, you can get insights into mass accuracy, isotopic patterns, curve fitting, and signal to noise. And this is all fully automated. So you can nicely see here some color coding. In green are all the glycan compositions that meet all the criteria that we have set for these specific features. So here you can see in blue dotted line that is actually that we said, okay, we allow a ppm accuracy of a ppm error of 10 and minus 10. But as we nicely have a look over here, we can see that most of the analytes that fit all the criteria fits within this 10 up to 0 ppm error. So you could actually narrow down your search a little bit more. On top of that, you could also look at your isotopic fitting. So how does the measured isotope compare to the theoretical isotope? What is the signal to noise of your compositions and how is the curve fitting? So it nicely visualizes, and also from this you can really see if there's a systematic error in your data or whether or not you still might need to do an extra calibration on your file. So how does it work? We have in GlycoGenius, if you would open it, you have a specific workflow. Here you select your files. You can add your files. So these are MZML or MZXML files. And once you have uploaded those, you can set your parameters. And the parameters include generating your library, but also, for example, how to perform your analysis. So if you focus on the library, we have some specific libraries that are already in there. So you can select whether you have an N-glycan, O-glycan, or a GAC sample. So there we make some suggestions what you could use. But if you want to explore a little bit more, what you can do is then just select, okay, how many monosaccharides do you expect in total? How many fucoses, sialic acids, etc. So you can make your library as big as possible. So you can incorporate, for example, that you're looking for N-glycan, so it will have a little bit of a specification that it will at least search for the specific SIDO bio score. But if you would not allow it, it will just generate a whole composition library. But of course, the more features you have, the more compositions it will search for, and also your search space will get bigger and will take more time. You can here add reducing end tag. So we have some most common tags incorporated. If you do permethylation, reducing end, or if you maybe modify your sialic acids, you can also click that on here. Whether you're looking for protonation or sodiation, all of this can also be added. How many charge stages you maximum are looking for, whether you're measuring in negative mode or positive mode. And then here also if you have sulfation or phosphorylation that you expect. Then if you want to do a fast isotopic distribution, we would recommend this. If you would have, for example, a measurement with an FTICR, you could do the high resolution distribution calculation, but then it takes again a little bit longer. Then from here, you can say, okay, how many CPU cores should be used? And here you specify what is actually the accuracy of the system that you measured on. So we normally measure on a QTOF. So here we say 20 ppm is still fine. But if you're messing maybe with an Orbitrap, you would like to lower that. On top of that, you can also specify the retention time or migration time range. So if you have a full analysis in our CE data, normally we actually only have maybe a 10-minute separation window, but we have a measurement of 60 minutes. To minimize your search space, again, you can specify where to look at. Then here you can also say, okay, how many minimum data points it should have per peak, limit the peaks picked per extracted ion lactoferrogram. So how many isomers, for example, would you expect? All of this you can specify, and then you can save your parameters to a file. Another option regarding the library is that you can also use a custom glycan list. So here, if there's anything that you're specifically looking for, or if you know you're looking at immunoglobulin and you only expect the top 15 glycans, or you only want to integrate the top 15 glycans, you can also use the one letter code, put it in this list. You can also load it from an Excel file, and then use that list for further data analysis. Then after that, what I also wanted to show is, okay, what to do if you want to do calibration. So with this option, you can do internal calibration. In our files, we have a lot of sodium adducts, so we could use that to calibrate our data still internally. And then here you can see it will automatically search for these compounds. It will tell you where it has been found in your chromatogram, what the M over Z error is, and what the actual ppm error is. And with based upon that, you can say, okay, what fit do you want to have? Do you want to have a linear fit or quadratic fit? What is the search tolerance, as well as what is the minimum intensity that you would look for? And based upon this, you can calibrate your file, and you can do this for all your samples in a similar way. Then it's of course time to really look at our data. And this is something that you could expect. So here we have a veteran sample, and here you see all the glycan compositions that have been identified. Here you see the compositions quality, and we see that we have 111 glycan compositions that meet all the quality criteria over here. On average, that means that we have the yellow composition. So that actually means that one of the quality criteria has not been met. And if we have red, that means bad, and that means that we are not meeting two of the quality criteria that have been set. And also, please note that we have an ambiguity notification over here, because if you allow a lot of different glycan composition, there's a good chance that maybe with a slightly different composition, so if you change from the top of my head, what was it again? A fucose
A glycoleo and a fucose for exos and
and a
acetylsalicylic acid. Yes.
thank you. Yes. And that would exactly result in the same M over Z value. So that will tell you whether or not there's an ambiguity and that you need to be careful. So that you maybe want to look into that, which glycan composition is actually most likely based on MS2, for example. So then if you, for example, click on this peak, you can also see, okay, but what is actually now measured? So here you can see the real values. What is the curve fitting score, the isotopic pattern? Here you see the theoretical one. So that is the red one. And then in blue is actually what has been found in our data. And also here you can nicely see your curve. On top of that, what is your ppm error? What is the area under the curve? And also, if there's MS2 available, it can also tell you how much of your MS2, so the total ion chromatogram in your MS2, is explained with this glycan composition. And if there are any ambiguities, it will tell you which other glycan composition is having the same M over Z value. So from there, now why is GlycoGenius actually really different? Well, we hope that we now have a fully automated LC or CMS data workflow. It's hopefully transparent. We at least try to make it as transparent as possible. So you can really look at your own data and see whether or not you agree with these assignments or whether you want to loosen your quality criteria a little bit more. It should work across platforms, so it is available for Windows, Mac and Linux. It's open source, and that also means that you can tweak the code or extend it. Ideally, we do it in collaboration because if you are looking for a specific feature, most likely someone else is also looking for that feature, and we would like to integrate that in our source code. And on top of that, it's very reproducible because normally everybody is doing this a lot of manual annotation. And in this case, we make sure that every time you're doing your data analysis with GlycoGenius, you get the same results. Just to show you some examples, this is an glycan analysis of total plasma and glycom. And this figure I made by myself manually using data analysis. And I did a lot of manual annotation on this data. I used 1 microliter of plasma, but from here I could identify 167 N-glycans. So what I've actually done is that I searched manually all the literature that is out there on plasma and what kind of glycan compositions were identified in plasma. From that list, I think there were around 500 N-glycans. I Every glycan I manually checked and looked whether or not it was meeting my migration time, it was meeting the ppm error, and from there I could say, okay, 167, I truly believe that they are present in my sample. However, what I was not able to do manually was, for example, to check whether the isotope was really fitting to what the theoretical isotope should be. So now we kind of analyze the same data with GlycoGenius, and this is for the same figure. So what you can nicely see as a sanity check is that we generate exactly the same figure as we generated with data analysis. However, this is now a lot quicker and done maybe in 1 hour, for example. And if we then look deeper into the data, what we can see is, as I said, I use literature to search for a lot of glycan compositions that have been reported in literature. And from these, I could identify 167. If we then use GlycoGenius, we could identify and see that 115 of the glycans that were found in literature as well as in the article were indeed found by GlycoGenius. However, a tiny part was actually not identified by GlycoGenius. So we went deeper into this, and actually what we've noticed is that one, not all compositions were detected in all replicates because we had replicates in these sets. And also, a lot of these compositions were actually not having the right isotopes, or at least they scored pretty badly. And also, there were a few compositions that were only found by manual search, and that meant that they were really hidden in the noise and that there were other isotopes kind of overlapping. So it is really, really difficult to whether, say truly whether that analyte was there or not. So it was interesting to see that now with GlycoGenius, you can do this very automated. I don't need to spend hours, days, or even weeks, I think I spent on this cohort to analyze. Then we also applied it on oak glycans. So we used an article that was recently published, and here you can also see that this glycan composition set was a little bit smaller. But overall, what we also did is that we analyzed the same data set with GlycoResolve because that is kind of a similar platform with some similar properties as GlycoGenius. In this case, we used very similar settings. And what you can see is that 16 of these oak glycans were found by all platforms, so also by manual interpretation using or supported by Skyline. Then if we focus on those that were actually only found by GlycoResolves, we could actually see that also here that those 7 glycan compositions were not meeting all our quality criteria that we have set. So that actually means that we are not fully sure whether this glycan composition is true because maybe the IPQ, the signal to noise, or the ppm or error was deviating too much in our opinion. Then on top of that, we also looked at a few that were actually identified by the article as well as by GlycoResolve, but not by GlycoGenius. And here again, we nicely see that these were all glycans that were not fitting our IPQs or isotopic scoring that we had actually put. So we normally look for an isotopic scoring that is above zero point 0.8. And I will come back to that later, why we feel that that is the right scoring to put there. And this is really nicely showing that with GlycoGenius, we can nicely visualize what is going on, but also why certain data might not be fully integrated or is not picked up by our tool or not seen as a good fit. Then to the visualization. So this oak glycan dataset was visualized with Skyline, and as you can see, the data is a little bit messy. So GlycoGenius is able to kind of smooth your chromatogram, but also to filter out the noises that are present in your data. Then also here again, if you click on the peak, you can get more information on how does it fit with your theoretical isotopic pattern? How is the curve fitting? How is your ppm error? And on top of that, you can visualize and say, okay, I want to have glycan cartoons present on or in my figure. And here you can click on it, and then actually there are a lot of different compositions present, which you could say, okay, maybe this is the most likely structure that I expect. Then also here you can see your the distribution again, the ppm error, the signal to noise, the curve fitting score, and isotopic fitting score. And you can nicely see that how our criteria are fitting and which compositions, for example, over here, we feel that there might not be the real glycans that we're looking for. Then about MS2, because also that is something that we do cover in GlycoGenius. And so what we cover in GlycoGenius is mainly the one letter codes, and we say, okay, this fragment, for example, fits with a hexose, two hexnax, and the T means the tag, so the reducing end label. As well as, for example, here is an extra water. So it will tell you which of these ions are actually fitting with the glycan composition that has it that it was assigned to. It's not fully there yet, but we do feel that it does provide with some preliminary MS2 annotation. It gives you some extra information, and it also tells you like how much of this TIC, so this total ion chromatogram, so all of these ions, how much of these ions could explain this glycan composition. And if you have ambiguities, so for example, the N-glycollyl versus the N-acetylneurominic acid with the fucose and the galactose composition, there you can really more see in depth, like, okay, do I see oxonium ions that fit with the N-glycollyl or does it fit with the N-acetyl? And that is also something that MS2 or GlycoGenius can help you with. Then, as I said, I also wanted to tell something about, okay, why is the IPQ at 0.8 the best setting? And that is because there we don't get any false positives. So we explored our data sets, so the N and the O glycan data sets that I just showed. And there we assessed, okay, how does our isotopic fitting score influence our identities or identification? So you can see what the sensitivity does, the specificity, the precision, accuracy. And what we notice is that if we put it at 0.8, actually we don't have any false positives. And that is actually something that we are looking for. We also compared it to GlycoResoft, and also over here we see that in with this data set, GlycoGenius scores better than GlycoResoft in regard to the true positive rate versus the false positive rate. Then very briefly, we also have analyzed this, used GlycoGenius for the analysis of glycosaminoglycans. So these were disaccharides. And well, actually, that's not fully true. So these are tetrasaccharides and with six
No,
monosaccharides.
this is actually the disaccharide, but with
It is
different
the, okay, sorry.
sulfations.
Yeah, it's the
It's
disaccharide with,
the
yes, the
yeah.
position of the sulfation. Sorry. Yes. And then here you can see it's with a 2AB label. And also over here, we looked, okay, what has been reported in the paper. So we noticed that, okay, there were a lot of different compositions reported. But then there were only 24 of those that we considered to be good in GlycoGenius based on the IPQ and all the other parameters that we have set. And then actually those that were not detected by GlycoGenius were related to either the ppm error was set too high, they were mixed with contaminant peaks, or they were actually not found by us when we did the manual search. So we are not really sure if they were really there. So this really provides a quick insight whether or not your glycans are there. and we hope it's really beneficial for you. So why should you use it? So I hope I could show you that you can go from raw data to really annotated data. You can identify but also quantify your data. And this is not only applicable for N-glycans, but it should also be applicable for O-glycans, glycosphingolipids, glycosaminoglycans, and if you have, for example, a glycopeptide where you only have a single peptide backbone, so something like immunoglobulin, so IgG1, then it's also fitting very nicely because you can put your peptide backbone also in GlycoGenius. So we really feel that, at least we hope that this is not only for us, but this is for the whole glyco community because we feel that everybody has been struggling with data analysis, and we really hope that GlycoGenius can be salvation for this. And on top of that, if you generate your data with GlycoGenius, after that, you could, for example, directly load your file into Metabo Analyst and get a very quick visualization of your data. So to wrap up, it's fast. You get confident MS1 annotations. It's early states in MS2, but you get full transparency. So you can exactly see why a match was made. You can tweak your output. You can select how your parameters should be set. If you don't fully agree that zero point 0.8 for your isotopic pattern might be a bit too tight, you can loosen it. But at least you're treating your whole data set in the same way. So behind the scenes, of course, I need to give a huge thanks to Hector because he not only wrote the entire pipeline in Python, but he also really designed the algorithms how to do everything. So everything has been written in Python. There are more than 20,000 lines of code. And it's really amazing what Hector has been able to do with this. And he will be able to tell you a lot more on this. So what will we do in a hands-on experiment? So we will do the workflow. You will select your spectra files, set your parameters, generate the library, then run your analysis, of course, review and filter your results, and then from there, export and visualize your data. And what you will hopefully learn from exploring this tool is that you can run a complete glycomics analysis from raw spectra to final data tables. You can interpret your quality scores, but also how to tune your thresholds, visualize your results with glycan cartoons, and then in the end, export your data, for example, in tools like Metabo Analyst to really further explore and visualize what's going on if you have different groups in your data. So GlycoGenius, it's open, evolving, and really built for the community. So we really would like you to try it, to break it, and hopefully with that, together we can improve it. So with that, thank you for your attention. This was the short introduction about GlycoGenius. And next up is to really do the hands-on experiment. Thanks. If there are any questions, I would be happy to take them.
Any questions? We can take a break in between if you want to ask any questions.
I think that's a good idea for the meeting itself. I have a bunch of questions, but I would say let's do the hands-on first, and maybe some of
Yes?
them get resolved.
Yes,
Okay.
that sounds good. So do
Okay.
you want to have a short break? I don't know if everybody was able to download the tool and the files. I saw
Um,
that Stephanie just popped in, so probably not yet.
Yeah, sorry about that. My kid was
No,
not
it's fine.
cooperating.
No, that's completely fine. It's I put in the chat, but I don't know if you still see it because you entered a little bit later. Um, so I will put it in there again. So on GitHub, can find the software tool, and then on the Google Drive, we put some MZML files that we would like to try for you to
Yeah.
explore the software tool.
One of the first things is whether everybody's working on Windows or are they
Mm.
using Linux or Mac. So for Windows, we have a executable file, which might, as Hene mentioned, be detected by your antivirus, but it's safe. It's just because it was built from Python. Um, but you can run the Windows file directly. But if you are using Linux or Mac, you should install it through the Python package manager from the repository for Python, which is PyPI, by typing in your terminal a command. I don't know if I can share my screen, which has also the codes. Wait a second.
So is anyone working on something else than Windows at the moment?
Yes, I am on Mac.
Yeah, I
So
would say
Yep.
most of the handout instruments for the handout laptops will all be Windows. A lot of our other software, so for example, some of the glycopeptide work and then the other glycomics stuff, most of those are very Windows focused. So I would say probably 90% would be on some kind of Windows.
Yeah.
Renee, do you
yeah,
think that's fair for your end too?
Yes, I think that's fine. We cannot really control the notebooks that people will bring.
yeah.
Since we are already on that, I looked at the GitHub. There is a short instruction for PyPy for Mac users. Maybe you want to add something like a link about installing Python first. If
Okay,
there is
I
like
can
a
see
web
that.
page, because I don't expect, a lot of people to be bioinformatics people. They are more like biologists and, chemists that are really analyzing the data. And installing Python can already be a challenge.
Okay.
Um, you know,
Yeah.
No,
but
that's good.
I actually don't have a Mac to test, but I don't know
I
if
see.
someone from your lab has it. Guinevere, you could also do some tests if I
No, I
provide you a
don't.
tutorial. Yeah.
Well, I can ask my husband. He has a Mac. I don't know if
Yeah.
his company allows it, but I can ask him.
Yeah.
Well, we have
I will see also.
we have a few Mac notebooks. We can try it on that one.
Yeah, that
But
sounds good.
as
I mean,
if somebody goes directly to the repository to install it from there, like I said, the instructions is a little bit short
Yeah,
for
yeah.
non-experienced
sounds
people.
good.
So if you can add some more material, that would be nice.
Okay. That's fine.
Okay, so I looked a little bit at the time. We are around 30 minutes right now, or
Hmm.
Jennifer's talk took around 25 to 30 minutes.
Yeah.
Um, we will be a bit close on time, so you will have a total of 1 hour exactly.
Okay, then I will shorten
Because
it.
yeah,
Then
it's the last session, so people will be leaving afterwards because
Oh,
yeah,
okay.
plane is waiting or not waiting. Um,
yeah,
so we cannot really, and you want to also allocate. I mean, it depends on how you do the hands-on. Usually what I do is, like I said, I show it on the screen myself, and then I give people 5 minutes or so to just repeat the step, do it
yeah.
themselves based on
Yeah.
the handout. And then usually during that session, while we are doing things, we are also answering questions.
Yes.
So you
Yeah.
don't necessarily need a very extended Q&A session in the end because you can resolve most of the questions
Yeah.
while doing it.
Yeah.
Sounds good.
And
Um,
also, you feel free to ask anything during the step-by-step
Yeah, yeah,
tutorial, and we can
mm,
also try our best to answer it.
yeah. And like I said, we will right now take a look at the software. How useful we will be in terms of helping people, we will see. But worst case, I will just say here, can you repeat that? Or somebody
yeah.
has this question, and we will
Okay.
That
see
sounds
how
good.
that works.
Yeah.
Okay.
Sena, were you able to get it installed on your Mac?
Stephanie?
No.
I didn't have Python, so
I'm
I'm working on it.
Okay.
Oh, yeah.
I'm
So
working
that's
on it now.
one
Okay.
issue to
Yeah,
to
to
resolve. Yeah.
resolve, definitely.
Yeah.
Yeah. But why don't we go
But
ahead
Yeah.
okay.
with the demo, and we have the recording later on in case somebody wants to look at
Yeah.
To
it
follow it
again and
again.
do it on their own notebook.
Yes.
Perfect. Okay, so that's very nice that you wanted a very step-by-step and take a minute to do your step presentation kind of tutorial, because that's exactly what I did. So you all can see the screen well, I guess.
Yes.
So the complete workflow that we'll follow in this tutorial is divided into 6 steps, and they are all presented in the user interface. So step number 1, select files, then set parameters, generate a library, run the analysis, check your data, and then at the end, save your results to files, export your results. So we'll go step by step on this tutorial. So for the first step, we'll click on select files. This will open this window, and you can add raw spectra files to your analysis by clicking on the add file button. It will open a Windows dialog, and then you can select your files all at once if you want. And they will be added once you click on open. They will be added to that list, where you can also remove any files you may have accidentally added, for example. Afterwards, you can allocate them as you wish. Should I go ahead? I think I'll go ahead. One of the things that you can also do on this window, if you have multiple groups to analyze, this might be important later on. It will save you some time later on, is that you can use this arrange groups button. This will open a window where you can add new groups to the top and you can name them, for example, control and treated. And what you can do is that you can just click and drag your samples from the bottom area of the samples into each of your groups. And then you can press OK, or you can save the groupings to files if you want to load them later into this automatically without having to drag everything again. But since we only have 4 files for this tutorial, 2 for each group, it might not be possible to use it on a table analyst since it demands 3 files per group. So this is more something for you to know later on if you want to analyze different groups. But once you have loaded all your files, you can just click on OK. It will take up to a minute to load these files, perhaps 2 minutes, depending on the computer. And then we'll get back to the main window. And you will see a green rectangle around the select files button to indicate that you have completed that step in the workflow. So here on the main window, once you load the file, if you want to explore your raw spectra file, you can also do it by just selecting the file here on the top left side. And then you can click on base peak chromatograph and click on any of the retention times to visualize that spectrum. But it will be raw spectra, so it will be really hard to see anything relevant at this point. So we move on to step number 2, to the second step in the workflow, which is set your parameters. You can start this step by clicking on the Set Parameters button at the top, and then it will open a new window containing all the parameters that we have to set. And this step is basically based on the properties of your sample, how it was acquired and how it was prepared. And here I provide you some of the examples for the tutorial files. So these are N-glycans released from mammal cells protein extract. These are derivatized with procainamide reducing N tag. These were analyzed in a QTOF MS with a typical accuracy of less than 20 ppm. And it was acquired in positive mode. So how does each of these features, these properties of the samples relate to the settings? So for the N-glycans, for example, you can click on the standards library parameters and select N-glycans. This will already adjust many of the parameters for the library building. But for this tutorial, to keep it a little bit shorter, we will reduce the maximum number of total monosaccharides to 12. The second feature of the sample, the second specification of the sample will be adjusted on the reducing N tag. You can check the checkbox and then select from the drop-down menu Pro A, which is the procainamide reducing N tag. For the fact that it was analyzed on a QTOF, it means that you have to adjust your accuracy appropriately. So you can change the accuracy unit to ppm and adjust the value to 20. If you want to adjust it to a higher value, it will also work because you can filter out your features by the accuracy value later on. But for this tutorial, 20 will work just fine. And the fact that it was acquired in positive mode means that you have to set the IO mode to positive here. The rest of the settings can be left as default, except for the retention and migration time range. We will adjust for this tutorial to 20 minutes. I provide optional workflow also on the written material. I can show it during the analysis how it works. But you can also, if you have no idea where does your glycans elute from your sample, we also have a small workflow to determine that and help you with the analysis. So for now, we'll set it from 1 to 20 minutes. And then all you have to do is set a working directory. Will be the folder in your computer where all the files created by GlycoGenius will be saved to. And then you can just confirm the parameters window. And once back to the main window, we'll again have a green rectangle around the set parameters button, indicating that you set everything all right. If it's red, that means that you probably didn't set a working directory, which is an essential step to make everything work correctly. So if everything is green to this point and everything has been set up correctly, we can go to the library generation, which happens by clicking the Generate Library button in the main window. This will make a pop-up, a new window pop-up, where you can set a name to your library file. Perhaps you want to use this library later on for a different analysis. So you can set the library name here. You can also do those brackets, those date and time surrounded by the brackets so that it will automatically set the current time or current date into your data, into your file name. And then you just click OK. It provides you a default name as well if you don't want to mess with that at the moment. So you can just click OK. And then it will pop up a new window where it will start the library generation. For this library, it will probably take just a few seconds to generate it. For very large libraries, or if you are using a high resolution isotopic distribution, it might take a while. So even for up to 10,000, it usually takes up to a few minutes with fast isotopic distribution. Once the library generation is finished, you can click on OK. And the button should turn green as well, indicating that you have done this step. What you can also do in the future is that you may also import an existing library by clicking on the Import Library button, or you can check the information on the library that you have loaded at the moment by clicking on the Check Info. This will open a new window. I don't have a screenshot of it here, but it will show you all the parameters by which the library was built on. And you can also browse the compositions inside that library by clicking on the Browse Library button. You can also generate files that you can use on Skyline, for example, to draw your EICs there from the file you generate here by clicking on Check Info. And if everything is done to the step, so you should have these three buttons green, we can move on to the analysis itself. So you can click on Run Analysis. This will pop up a new window. It's a warning asking you whether you calibrated your samples or not. This is more of a reminder. If you didn't calibrate your samples, you can calibrate them. You can cancel and calibrate them. But the files I provided are already calibrated, so you can just click OK. Then set a name for the results file. You can do it in the same manner as the library file. Then click OK, and this will start the analysis. So it has a log that displays at what step it is. It has a progression bar as well. And it should, for this analysis, it should take between 5 to 10 minutes, depending on your computer specs. And during this time, I can also show you some other things while it's running on your computer, if you wish. So this is a good step to ask you how is everything going. I don't know if any of you are following step by step. René, perhaps, or Melody?
Yes.
Yeah, this is working fine.
Okay. So in this time, during this 5 to 10 minutes, I was talking to Guinevere. One of the things that I can show is how you can find out what's the retention time that you want to analyze. So if you allow me, I can also show you in the meantime. This is also provided at the end of the written material as an optional workflow if they want to do it themselves. So let me show you. I will share another window. And please tell me when you are done with the analysis so that we can also move on. So I'll share my whole screen, I think, at this moment. Yeah. So you should be seeing my whole screen with GlycoGenius open.
Yes.
Okay. So for example, I will add one of the files that we use in the tutorial. It can be this one. So you load the file, the spectra file, and here on the top right side of the window, you have this button here, which has a square button with a drawing of a spectrum. If you click on it, it will process your sample and create a single spectrum for your whole chromatogram. So we call it the maximum intensity spectrum. It will bin all your spectra into MZ ranges, and it will take the maximum value that each M over Z value achieves and create a single spectrum for that sample. And you can interact with it, zoom in, but you can see the binning if you zoom in a lot. So if you have this window open and you click on the import library, oh, I don't have a library generator. I can generate it very quickly. So ProA, let's put here on 12, positive mode, save it here. Okay, generate a library. So if you have your library loaded and you open this window, you have this button on the top right side of the window. It's called Quick Analysis. If you click on it, it will instantly identify from your library all the glycans that were found within your sample on this spectrum. You can arrange them by the isotopic fitting score by clicking here on the top of the header. And you can also click on each of the glycans to see the isotopic envelopes if you want. This is kind of a rough analysis of your sample. But this analysis is not just detecting the M over Z values, it also scores the isotopic fitting, so we can use that. Once you have done this, you can close this window and you can click on this square button here on the top right side of the main window. It's the quick traces window. And here you can type in on the M over Z values, MIS. If you do this, it will automatically load all the M over Z values that were identified in the MIS window, the maximum spectrum intensity spectrum window, in the same order that you order there. So, for example, if I click on the first spectrum here, or on the first M over Z value, it will draw a raw chromatogram of that M over Z value. So, for example, if you hold Shift and select 20 of them, or so, I don't know if I selected 20 at this moment, it will draw an EIC of every one of them. So if these are the glycans with the highest confidence in identification on that rough analysis that we did, it should trace glycans into your chromatograms there. And with that, you can easily see the retention time range at which your glycans eluted. So this is a very quick way to see what's the retention time or migration time range that you want to analyze. Within less than 5 minutes, you can determine that. And that will, if you set the correct range there, it will really shorten the analysis time. So for our tutorial, the files might take 5 to 10 minutes. But if you are doing a big data set, so for example, 60 files, and you have a library of 1,500 glycans, it might take many hours to run it. So if you can restrict the retention time and or migration time window, it really improves the runtime of your analysis. So
Can
as
you
you can
zoom?
see here,
Oh,
sorry. Oh yeah, I can zoom in. Let's just wait for the tracing to finish. But as you can already see, as they are showing here, most of the glycans, they are in between. So here, it's done. So you can zoom in, as you can see, and most of the glycans are between 0 and 20 minutes, perhaps. We can do a little bit more just to be sure. So we have a few peaks here, for example, at 16 minutes. We have peaks, most of the peaks are here between 0 and 5 minutes, and nothing is behind the 2 minutes. So we can set a window from 1 to 20 minutes, which is the one that I provided for you in the tutorial. But if you ever if you ever find a sample that you have no idea what's the retention time, you can do this small workflow to determine the best range of retention time to analyze. Okay? So I imagine that by now your analysis should be close to finished.
At least on my computer, it's done.
Yeah. So the more CPU threads you have, the faster it will be. So it can use up to 64 threads. On my computer, it has 12 threads. It takes roughly 5 minutes, 5 to 6 minutes to run, for example. So I think I can move on. During the analysis, if you are, let's say your analysis has been running for a while, or you have some doubts about some of the settings that you've set, you can click on the check analysis parameters in the running analysis window, and it will also provide you all of the settings that you set during your analysis and which files you are analyzing, so that you don't lose track of that. Okay. Once the analysis is done, you can click on OK, and it will go back to the main window, but this time it will load all of your glycans on the left side panel. So the next step in the tutorial is to check your data. It's even though everything is automated, you want to set the best quality score thresholds as possible for your data. So you have a lot of tools here to properly check your data. I will show you one by one. So you can choose a sample from this drop-down menu on the top left side of the window. You can filter your glycans, your identifications list. So for example, you can type in H2 to find all the glycans that have 2 exoses, for example. You can type in also the M/Z values of glycans if you want to look for that. Here you have a list of all your identifications, and you can select a glycan, its adducts, or its peaks to see their chromatograms. And you see that it's color-coded. So as Guinevere said earlier, if it's green, it means that it's a good glycan. It meets all the quality thresholds that you've set. If it's yellow, the best peak within that glycan identification failed exactly one threshold. If it's red, the best peak failed 2 or more thresholds. There are ways to check each of the scores. Another thing that you can check to properly set your thresholds is to check the score distribution, as Guinevere showed. And you can also set the thresholds here in the main window. And if you change the thresholds, the colors and the quantification of the good, average, and bad glycans, it will change in real time. Also, the quality scores distribution, it will also change in real time. Everything will change in real time, so you can kind of test and check how it runs, what's the best setting for your sample. We recommend a 0.8 of isotopic peak thresholds. The curve fitting score is more important for quantification, so that relates to the shape of the chromatogram peak. Signal to noise ratio is also an important parameter. If it's too low, the signal to threshold ratio, all your scores for that peak might be a little bit lower than usual. And the ppm error is mostly related to your MS analysis, so it should be tied to a specific range based on the equipment that you used. You can set a different boundary for the inferior and the superior limits as well. So you can visualize every chromatogram that you click on the left side panel on the chromatogram viewer on the top side. And if you select any of the rotation times in the chromatogram viewer, it will display the spectrum. And if you select a peak on the left side panel here, if you select a peak, it will show the boundaries of the peak here on the top, as you can see on the chromatogram viewer. And if you click on any of the data points within that peak, it will also show the peaks that were chosen by GlycoGenius, that they were considered the correct ones. GlycoGenius, during the analysis, it already filters isotopic envelopes that don't fit the target glycan. So for example, with different charge state, for example, or if it's not a monoisotopic M/Z value, it filters that out, but you can also check that by clicking on the chromatogram. And what you can also do is double click peak retention time or migration time on the list, and that will open the peak analyzer. On this window, you have the chromatogram of the peak at the top. And if you click on any of the data points, it will also display the isotopic distribution of that peak. And you have in red the theoretical one or the ideal one for the chromatogram, which is a Gaussian for the chromatogram. and a simulated one for the spectrum at the bottom. And you can click on each data point and see how each data point scores for the isotopic fitting and how it should be the ideal one. You can see all the scores here on the right side and also the area under curve. And if it has any ambiguities, you also see the glycan with which it has an ambiguity, so that you can properly see why a given glycan failed a quality check, for example. What you can also do is click on this button here on the top right side. It's the GGDraw module. That will display the glycan cartoons on the chromatograms. It might take a few seconds to load the first time if you only have one chromatogram, because it first has to draw the glycan in your machine. So it takes a few seconds the first time. And then after it's done, it will generate the cartoons, and you can click on the cartoons, double click the cartoons to also select a different cartoon if you think that's more appropriate for your glycan. It's to note that at the moment, it doesn't choose a specific one. It's a random one. So it should be based on your knowledge at the moment. But you can select a different one and click on change glycan, then close the window. It will change there. You can also drag around the glycan on the chromatogram if you want to rearrange that for a better figure, for example, for a paper. Or you can also change the size of the glycans by clicking on this dial here on the top right side besides the GGDraw module button. So once you checked all your data, you can export the results to files. So just by running the analysis, just by finishing running the analysis, you already generated a GG file. That file contains all the identifications that you can interact with just as you are doing right now. So you can load all this data later on if you want. So that's already saved. But if you want to export data tables, for example, from your data, you can click on the Save Results file button. That will pop up a new window that looks like this. It already has some default settings. If you leave it as default, it will use the thresholds that you set on the main window, and it should generate one result file that is an Excel file that should have many sheets in it. So basically, the first sheet is an index of all the MS files that you analyze. So for example, it has a sample number, the file name, the average noise for that given file, the number of identifications in MS1 or MS2, if you have MS2 analyzed as well. It already does some, it already divides your N glycans by subcategories. So fucosylated, sialylated, fucosylated and sialylated, Pulse-Mannose, hybrid, Ham-Mannose, and complex. On the second sheet, you have the complete identifications list for all your files. So this will display all the identifications, whether they were identified as singly charged, doubly charged, triply charged, and so on, and on which samples they were identified that way, and the number of samples they were identified. And for each sample file that you provide, you also get a table, a complete table of identifications. This one will show the glycan, the adduct, the M over Z value, the retention time, because the same adduct might have multiple peaks. So that can be different isomers for glycans, for example. It will show the area under curve, which is the abundance of that specific feature. The scores, the isotopic fitting score, the curve fitting score, whether it has an ambiguity or not. If you analyze the MS2, it will also provide you the total ion current annotation as well. And it will also provide a list of of all the adducts combined for each peak of each glycan. So for example, you might identify multiple peaks for a single glycan like the H4N2 here. And it will put together all the adducts for that. So this is kind of the convolution. And also it will provide compositional separated information. So this will take all the peaks for a given glycan and combine that into a single composition identification. So for here, we had 2 peaks for H4N2, and these 2 peaks area under curve were combined into a single feature here. you can also output other files. So for example, if you click here, you have a unified table with the abundance of all your identifications already filtered by the quality criteria thresholds you've set. And it provides files that are peak separated, composition separated, and they are also group separated if you set the groups. It will look something like this. The idea of this table is it should be easier to pipeline this table into other scripts if you want to follow up with a different workflow afterwards. So this already provides you the abundance of every glycan already curated by your quality criteria and separated by groups, as you can see here on the top. This file can also be directly used on MetaboAnalyst, which can already do some grouped analysis on your sample. So you can quickly generate PCAs, heat maps of your identifications. You can do t-tests or ANOVAs of your samples and try to find out if any specific glycans, for example, might have already given you a significant different value, for example. You can also output a file containing all the chromatograms that were identified, the data points for all those chromatograms. If you want to plot that in a different program or something like that, you can use this button. It might take a little bit to generate this file because it's a lot of data. So I don't recommend using it unless you really need to. And after you are done, you can basically confirm, and then you have all those files to analyze. It's to note also that you can right-click any of the plots in the program. So for example, the chromatogram or the spectra viewer, and save an image of that. And you can also save an SVG image, which is an editable vectorized graphic file. So for example, you can import that into PowerPoint, for example, and separate the elements and change the colors or do whatever you want with it to make it look better if you want. So that already helps you a little bit in creating article quality figures. So I recommend you to try out the other workflows found at the end of the written tutorial yourself, and also visit our Trello dashboard if you want to check out the intended features that we have for the future, or if you want to report a bug or suggest new features, you can feel free to do that. And I think that's all for the day. And if you have any questions, I'm up to answering them. Oh.
Okay. Let me stop the recording.